This is the lecture for Chapter 14, Disease and Epidemiology. First thing we have to do in this chapter is to go over some basic vocabulary. And actually, this chapter has lots of vocabulary for you to learn. First term you should learn is pathology. Pathology stands for the study of disease. And patho originally relates to suffering, which is what happens when you have a disease. Second word is etiology. The etiology is the study of the cause of disease. So what is actually triggering that disease process in an individual? And the third term is pathogenesis. So patho, disease, genesis, to begin, and to propagate, so how the disease develops. Now the next two terms are terms that are often used in regular conversation interchangeably, but they are not interchangeable. The first of those two terms is infection. An infection specifically means that an individual has been invaded by a microbe. So it means an individual has been invaded or colonized by a microbe, so that microbe is living on or inside of them. The second term is disease, and disease basically means a change in the state of health of the individual. So it means that some part of their body is not functioning normally. So it's a change in their state of health. So you can have diseases that have nothing to do with infections. Heart disease, genetic disease, all of those have nothing to do with being invaded by a microbe. And conversely, an individual can be invaded by a microbe or colonized by a microbe and not show the disease. It could be they are immune, or the disease is latent, it's not expressed yet. In this class, the disease that I will often be talking about is infectious disease. So in this class, I'm not specifically going to say infectious disease all the time. I will probably just use the word disease, but I'm referring to diseases that are caused by being invaded by microbes. So that is infectious disease. When we first start talking in this chapter about the different microbes, different types of infectious disease, one of the first things we want to go through is your normal flora. So your normal flora, those are the microbes that are living on a healthy, normal person, living on the inside and outside of that person. And I think I mentioned before that you have 10 times the number of microbes living on and in you than you have actual cells. And if you took all those microbes and took them off of your body and weighed them, they would weigh about a half pound. So you have a half pound of microbes living on and in your body. Now, where did that flora come from? Well, originally, when a fetus is in the uterus is a sterile environment. So when you have a fetus in the uterus, there's no flora present on that baby. But as soon as the baby starts going through the process of birth, then the baby starts acquiring microbes from interacting with the microbes in the vagina. Then when the baby is born, inhaling air. Air is not sterile, so the baby acquires microbes that way. And then of course there's food. Any food the baby takes in, again, is not sterile, so the baby is acquiring microbes that way. And any time the baby is touched in any way. So all of that is where you started acquiring your normal flora. The next few slides show where flora is located on a normal, healthy person. And the first place, of course, is your skin. So you are covered in microbes all over your skin. So the yellow here shows where the microbes are. And they aren't just on the surface of your skin, but they're down in the hair follicles, they're down in the glands, and the ducts of various glands. So your flora is deeply embedded into your skin. Also in the digestive system, a normal healthy person will have tons of microbes in the upper part of the digestive system. And already I've mentioned that your mouth has the highest density of microbes. And then also in the large intestines. So at the two ends of the digestive system is where a healthy, normal person has flora. Respiratory system, a normal, healthy person will have flora in the upper respiratory system, so that includes the nasal cavities and also the trachea, all the way down the trachea, but not into the lungs. So again, a normal person would not have normal flora in their lungs. The last place you would have flora is in the urogenital system, in the distal end of the urethra in both the female and the male, and then also in the vagina for the female. But normal flora does not go into the uterus, not normally found in the uterus. And again, when there's a fetus in the uterus, that fetus is in a sterile environment. When two organisms are living in close contact with each other, such as a human with their flora, that is called a symbiotic relationship, so symbiosis. And symbiotic relationships can be divided into three types. The first type is commensalism. So commensalism is when one organism benefits and the other is unaffected. And in terms of your flora, this happens a lot. Often the microbes that are living on and in our body benefit, and many of them have no effect on our health or our body. Mutualism is the second type of symbiosis, and this is the situation where both organisms benefit. So both the microbe and the human have a benefit. The last one is parasitism. Parasitism is where one organism benefits at the expense of the other. So in this case, sometimes the microbes benefit and it can harm the humans. And when that happens, that's considered the development of disease. At the beginning of the semester, I went through the benefits of your normal flora, and just as a review, it helps for the normal development of your immune system and digestive systems. Also, there is vitamin production. I mentioned E. coli producing vitamin K in the large intestines. And the last major benefit of your normal flora is microbial antagonism. So the fact that your normal flora outcompetes potential pathogens. But not all flora is good. So some of the flora can actually cause disease. Again, the vast majority of microbes are harmless to us or even beneficial, but the few that actually can harm us are called pathogens. And these, of course, are the microbes that lead to disease. And remember when we talked about pathogens before, the two types of pathogens. First are the opportunistic pathogens. So again, these are the microbes that under normal circumstances, they're beneficial or harmless to us. But if they're allowed to overgrow or go to a different part of the body, then they do cause disease. They do damage our health. The other type of pathogen are the obligate pathogens. And these pathogens are obligated to infect a host, and in doing so, they will cause damage to that host. And the reason they have to infect a host is they need to do that to replicate or to gather nutrients. One particular type of flora is called transient flora. And transient flora is the type of flora that you pick up during the normal course of the day. So anything that you touch, you are picking up microbes from. Other people that you touch, you're picking up microbes from. And these are the microbes that are most likely to cause disease. Your permanent normal flora is less likely to cause disease than the transient flora. So what is the best way to protect yourself from disease caused by transient flora? It's to wash your hands. So that is one of the main things you need to do to protect yourself from potential pathogens that you are picking up during the course of the day. Germ theory of disease. So if you remember when we talked about the history of microbiology, I brought up Dr. Koch. So Koch, he was a big proponent of the germ theory of disease. And remember, germ should basically be looked at as the old-fashioned word for microbe. So microbe theory of disease. That was the idea where microbes can actually cause disease. And Koch was the person who developed a process in which to discover which microbes cause which disease. And this is Koch's postulates. So Koch's postulates, this is the process by which you experimentally prove which microbe is caused by which disease. So the first thing to do, this is a process that you need to know. The first thing you need to do is you need to find individuals that have a particular disease. And from that individual, you need to be able to isolate and identify the microbe that is found in that diseased individual. And the specific thing about this is that every individual that you find that has this disease, they must contain the same microbe. Once you have identified the same microbe that is present in all cases of the disease, then you need to take that microbe and inject it into a test organism. So you inject it or expose that test organism to that microbe, and then that test organism must develop exactly the same disease as the originals. So they must have exactly the same signs and symptoms, same evidence of disease. Once that test organism has developed exactly the same disease, then once again, you need to isolate and identify exactly the same microbe that you did in the first place. And this is how Koch's postulates work. So as review, all individuals that express that disease, you must be able to isolate exactly the same microbe from all those individuals. And when you take that microbe and expose a healthy organism to that microbe, that healthy organism must develop exactly the same disease as the originals. And at the very end, from that test organism, you have to be able to identify exactly the same microbe.